said, my name is Ashley Kendall. I'm a student at Georgetown College, and this is the presentation of a research project that I was able to complete this summer. We studied the synthesis of boronic acid derivatives as potential inhibitors of the enzyme ArgE. So instead of just going straight over your head and going right into it, we're going to talk a little bit about what we're going to talk about as well. So first of all, I'm going to talk about the importance of bacterial infections and how that looks in the United States, as well as their increasing rates. Second, I'm going to talk about antimicrobial resistance and how that's affecting the way that we treat bacterial infections in the United States. Third, I'm going to talk about the bacterial enzyme ArgE and its importance in the bacterial enzymes. And then fourth, I'm going to talk about the ArgE structure and inhibition. And then finally, I'm going to talk about our boronic acid derivatives that we were actually able to synthesize. So first, there are 2.8 million antimicrobial resistant bacterial infections in the United States every single year. So that's a really big number to try to process and to think about in your head. So if you're a Wildcat fan, I want you to think about Kroger Field. So within Kroger Field, you can fit 2.8 million people in Kroger Field 46 times. So that's a lot of UK football fans. So actually between 2012 and 2017, deaths from antimicrobial resistance decreased from 18% overall and by 30% in hospitals, compared to in 2020, bacterial infections increased by at least 15%, depending on the bacterial infection that was present due to the COVID-19 pandemic. This is due to research shifting highly towards viral studies instead of our bacterial infections, and that's why our rate started increasing. Here's actually a table of data from the CDC comparing values of different bacterial infections. And as you can see, almost every single one of those compared from 2019 to 2022 increases, except our MRSA. But almost every single bacterial infection that's common and causes the most deaths in the United States increased in infection rates. So the question is, how can we stop antimicrobial resistant bacteria infection rates from increasing? And our answer is the enzyme ArgE. So what is ArgE? ArgE is a metallohydrolase enzyme in the arginine biosynthesis pathway of bacterial cells and serves as a potential target for inhibition. So whenever you look at this pathway, this is actually the arginine biosynthesis pathway in bacterial cells. You notice that our steps with ArgE are highlighted in green. So that will kind of follow through the rest of the presentation. So why did we choose ArgE to target for inhibition in bacterial cells? First of all, we've noticed its presence. So ArgE is present in all gram-negative and most gram-positive bacterial cells. If you're not a big biology person, gram-positive bacterial cells can kind of be thought of as a really thick wooden fence. It only has that one double phospholipid bilayer and has a thick layer of peptidoglycan on top. And then our gram-negative bacteria have two phospholipid bilayers with a layer of peptidoglycan in the middle. The way that you can think about that is kind of like a bulletproof vest. It's more thick, it's harder to penetrate, and that Kevlar, which is the peptidoglycan in between, is really protecting it instead of a wooden fence, which is easy to target. Second, the reason that we chose ArgE is that ArgE is only expressed in bacterial arginine synthesis pathways. So it is not present in the mammalian synthesis pathway, which is what humans and animals all have to produce arginine within their cells. Here's some images of our pathways. And if you zoom in on the arginine biosynthesis pathway in mammals, you can see that you can't find an ArgE enzyme in any of these. And mainly the enzyme that synthesizes arginine in mammals is this arginosuccinate lyase, and that's what synthesizes our arginine. So we do not have that ArgE step that the bacterial pathway does. So right here you can see that ArgE is the enzyme that takes N-alpha acetyl-ornithine and turns it into L-ornithine and acetate. And then that leads us into our third reason why we actually picked ArgE. So the products made by the reaction of ArgE are essential for polyamine synthesis, which is used in DNA production. And then the final step in the arginine synthesis pathway, arginine, is used in protein synthesis. So these specific products are one main reason on why we chose ArgE as our target for inhibition. Again, that L-ornithine is used in synthesizing the arginine within the pathway, and then the acetate combines with the carbonyl phosphate to continue your pyrimidine synthesis for DNA. So how can we target the enzyme ArgE for inhibition? Our first step is to actually look at the crystalline structure of ArgE. And so now you can notice the main structure of this enzyme is in blue, with two specific molecules on each side that are highlighted in pink. Those, if you actually So if you actually zoom in on this crystalline structure, this is the active site that you're seeing, and those two pink molecules are actually zinc. So there are two zinc molecules within the active site that cause it to be the metallohydrolase instead of your normal hydrolase. So how do we target the active site? First, we looked at the reactant structure. As you can see, there's some negative and positive charges influencing the attachment to the active site to where it splits from that N-alpha acetyl-ornithine into the ornithine and the acetate. And then we synthesize a para backbone of this structure to add on our amine additives right here. Right there, that's where our amine additives all went on our molecules. And then next, we also synthesized a meta backbone to just see which ones work better within the active site and which ones produce the most interactions. So here's our reaction mechanism for what we followed for every single molecule that we made. This first step, we protected the boronic acid using 3 or 4 formal phenyl boronic acid and a pinacol to protect the oxygens on the boronic acid so they did not have interactions with the other reactions that it followed after. Next, we added malonic acid to make a carboxylic acid, and then we added a carboxyl chloride in order to have a reaction with that chlorine to make an amine. And then finally, we deprotected the boronic acid using diethanolamine. So the reason that I don't mention much about these steps in between is because we actually had to go through so many to find something that worked. It was really hard to pinpoint one that worked for every single molecule. So we synthesized over 20, I want to say we're about at 25. And so it was very difficult to kind of place a specific reaction structure for all of this. Now, here's an example of those para backbone interactions. So this is used, synthesized using MOE, which is called MO technology, and it actually places your molecule within the active site and tries to find the best fit of your molecule within your active site. So as you can see, we have strong interactions here, both with our carboxylic acid and then also with the benzene ring with the histidine 355. And so one other thing that you can take away from this is the S score. So the S score down here on your bottom right is negative. That's super, super important whenever you're working with MO, because the more negative an S score you have, the better the molecule fits into the active site. So as we go through these, pay attention to those S scores and I'll make note of it. So the first one that I'm demonstrating for us today had our lowest S score, which means it's the one that's most likely to fit into the active site best and have strong attachments. And that is our para backbone with a paraphenotidine derivative. So that is just that extra benzene ring with the oxygen and the ethyl group on the end. So as you can see right here, we have strong interactions with this arginine 252, as well as our oxygen interactions with histidine 195, and again, the same interactions with our benzene ring. Now, one thing that I want you to note that's really important is the reason that we chose boronic acid derivatives is these deprotonated boronic acids have high interactions with our zinc molecules within the active site. So those zinc have positive charges, and whenever our boronic acids are deprotonated, they carry a more negative charge, so those naturally interact with one another. And then the fit of the two oxygens on the boronic acid fit perfectly into the active site to match with those zinc molecules. Next is an example of our para backbone with 4-aminobenzotrifluoride additive. So that just means that we have a 3-fluorine on the end of our benzene ring over here. And you can see these all have different fits, and the molecules all shift different ways in the active site. That's due to the MO structure determining and just trying to find which way it fits best, and also has the highest interactions. So again, you can see we have our interactions with our zinc, interactions with histidine, especially with the histidine 355 in that benzene ring, and an S score of negative 11.5. So this one is a pretty good fit as well. And then this one also has a S score of negative 11.5, which is interesting whenever you're looking at the molecules because they're so very different. This molecule actually has a chlorine that's interacting with isoleucine 351, and then pretty much the same with those interactions of the histidine 195 and the oxygen, as well as the histidine 355 and the benzene ring, and of course our boronic acids with the zinc. And here's another one of our para backbone with a orthophenotidine derivative. So the only different interactions here are this asparagine 251, and you can see those are really big blue bubbles, meaning that those show high interactions. Yet, our S score is slightly higher than the other ones that we've seen. So it all just depends on how these molecules fit into the active site and what interactions they're having that determine how well of an inhibitor they would actually be for our enzyme. And finally, we have a meta backbone with a orthoanisotine derivative. So I wanted to give you an example of one of our meta backbones because we actually saw that those do not fit into the active site as well due to their structure. But here's an example of one, and you can really see with that S score is positive 12.1. So that S score is saying that molecule does not want to fit into the active site at all, but it still shows the interactions that you're having with your orthoanisotine and your zinc molecules. So that's kind of how we decided which backbone that we were going with and which ones best fit. So now, how do we determine if these synthesized boronic acid derivatives that we've created actually will work as enzyme inhibitors? So the first test that our partners in Chicago looked at using was a 214 nanometer assay using high-powered liquid chromatography. And this is just a high-powered liquid chromatography that checks the final reactants of your enzyme reaction. So it goes through, and the reason that we chose not to use 214 nanometer based assay is because it's not useful inhibitors that absorb at UV, in the UV region of the light spectrum. So if you look in on this UV spectrum, you can see that our ultraviolet is right here at 10 to the minus 8, while our 214 nanometers is in the 10 to minus 9. So for all of our molecules that exhibit high absorption in the UV region, so pretty much anything with those benzene rings and those large structures, they're not going to show up in a 214 nanometer based assay. So we actually had to try out a different one that our research partners in Chicago created. So they chose to do a ninhydrin based assay because they were able to use the reaction. And what they did was they would run the reaction with the enzyme ArgE and then place it in ninhydrin. And ninhydrin actually causes an interaction between primary amines and the ninhydrin molecule, forming Grumman's purple, which is just a demonstration of whether or not there are still reactants in the reaction. So they would run a reaction, and in order to see how well the reaction was inhibited, or if it didn't work at all, then they would run this reaction, and if it turned purple, that just means our inhibitors are working. So if your test was clear, then that means your Grumman's purple is not formed, and therefore you don't have a primary amine anymore. You have that L-ornithine and the acetate instead of your original reactants. So this was used to detect the presence of reactants, and also because it treats the inhibitors that were not able to be used for the 214 nanometer based assay. So this is still happening for our molecules that are in specifics, but they do know that there are several that work as inhibitors. It's just not ours personally, so I don't have that data yet. It should be in on Monday. So I just want to go over a little summary of what I've talked about today. First of all is antimicrobial resistant bacterial infections are increasing in the United States. Remember, that's that 2.8 million infections per year that's been increasing since COVID-19. Second, that ArgE is a potential inhibition target of the arginine biosynthesis pathway in bacterial cells, and that's because of 3 reasons. It's because of its presence in cells, that it's present in all gram-negative and most gram-positive bacterial cells, that it's not expressed in mammals, which was that mammalian arginine synthesis pathway, as well as the bacterial arginine biosynthesis pathway, and that ArgE is the enzyme that functions in the bacterial cell pathway, and it's not present in mammals pathways. And then third is that our products are essential. So that acetate combines with the carbamyl phosphate in order to form our pyrimidine synthesis, which creates DNA. And then the final product of the arginine biosynthesis pathway, arginine, is used in protein synthesis. So inhibiting ArgE causes these cells to be unfunctional in their environment. And then fourth, our boronic acid derivatives may serve as potential inhibitors due to the interactions within the active site, both with the zinc and other interactions between the structure. And then finally, if our inhibition is successful, more antimicrobial resistant bacterial infections will be able to be treated more broadly, especially just due to the fact that ArgE is present in all gram-negative and most gram-positive bacterial cells. So I would just like to thank Dr. DiMaschino at Georgetown College for letting me work in this summer, as well as our partners at Loyola University of Chicago, Dr. Daniel Becker and Emma Kelly, and also for these students that worked with me this summer, Lucas Smith and Tara Bowman. Does anyone have any questions?
Questions?
I have a couple questions. So for the boronic acid derivatives, did you all do the MOE things before you synthesized them or after?
So some of both, actually. We had planned to synthesize some structures, and so we went ahead and sent that list to Chicago because they did all of the MOE in Chicago. But some of them we actually synthesized before and then sent it. So those are the ones that I have here, is the ones that we synthesized and then they make the structures for us in both.
Okay. And so boronic acids bind a lot of stuff, right? They could bind, if they're binding zinc, I'm sure they could bind any divalent cation, which are present in all sorts of enzymes. So is there like previous literature evidence or things like that that made you think that these boronic acid derivatives might be specific to ArgE and not just inhibit all enzymes and thus not be applicable to systemic infections?
So actually my research advisor, Dr. Dimschmino, as well as his partners in Chicago, worked with the DAP-E enzyme, which is in the lysine synthesis pathway of bacterial cells, and it has the very similar dizinc structure. And so they started looking into it further and found that ArgE was also a really good target for that because they didn't have as much success with the DAP-E.
Any other questions? Thank you very much.